Hi everyone, this is the 12th lecture for your chapter Cell. In the last class, we discussed chloroplast, okay, which is a type of plastid. It is the plastid which does the function of photosynthesis. It has double membrane. It is also semi-autonomous. Apart from chloroplast, we discussed two other plastids, which were leucoplast and chromoplast. We saw their functions and their structures. In today's class, we move ahead and we will be discussing the skeleton, okay, of cell. Have you ever seen a skeleton in scary movies or in the corner of your biology lab? Right. So skeleton is basically a backbone, something, a supporting structure, okay, something to stick your tissues to and to grow, to keep you erect, right? So same way, there is skeleton for cell also, which is called cytoskeleton. Cytoskeleton are extremely minute, fibrous and tubular structures which form the structural framework inside the cell. Okay, they are more of structural. Okay, they are structural support in the cell. They are not visible by normal light microscopy. Okay, you cannot look under a microscope and easily see the cytoskeleton. They are very small and fibrous. So, how do you visualize a cytoskeleton? It was only after discovery and use of fluorescent dyes and proteins. Okay, so now these fluorescent dyes, like red fluorescent dye or red fluorescent protein, green fluorescent protein, these different types of dyes and stains were used to stain a particular part of the cytoskeleton, let us say green, or uh, dye it red. Okay, and then by the phenomena of fluorescence, when UV light or light of lower frequency is sorry higher frequency is made to fall on these samples, okay, on these cells which have been stained, and you look under the microscope, you will see such kind of fluorescence. Okay, and then you can find out that okay, this blue colored one is the nucleus, the green colored ones are maybe, let us say, microtubules, the red colored ones are, let us say, intermediate filaments. Okay, so only after development of fluorescence were we able to see the cytoskeleton. Cytoskeleton only occurs in eukaryotic cell. It is absent in prokaryotic cell. They were discovered with help of fluorescence microscopy. The cytoskeletal structures maintain shape of the cell. Okay, of course, in plant cells and in fungal cells, they have cell wall for maintaining the shape. But who will describe it to the cell wall that where the wall should be? This is actually done by the cytoskeleton. Okay, so cytoskeleton maintains the shape of the cell. It regulates orientation and distribution of the cell organelles, where each and every cell organelle should be present. That is decided and maintained by cytoskeleton. For example, look at this diagram. You can see there are different organelles like this mitochondria, or like this, let us say, Golgi body. Okay, so some of these organelles should not move around. They should be fixed at some place. So this fixing will be done by the cytoskeleton. Okay, all these fibers that you see are actually cytoskeleton. They are of three types. You have microfilaments, intermediate filaments, and microtubules. These are actually arranged in their size, okay, order of their size. Microfilaments are the smallest in size, in diameter. Then comes the intermediate filaments, and finally the microtubules. We start with the microfilaments. They are long, they are narrow, cylindrical rods or protein filaments. Okay. Microfilaments are made up of actin protein, which constitutes 10 to 15% of the total cell protein. So if we consider, let us say a cell has 10% protein, okay, then out of that 10% protein, 10% will be microfilament. So 1% of the whole cell is made up of actin. Okay. If it is too complicated for you, just leave it, never mind. I was just calculating. Okay, so basically, if you take a cell and if you talk about its protein content, then 10 to 15% of the cell protein will be actin. Now, actin occurs in two states. Okay, it is seen in two states. One is called G actin. G stands for globular. Okay, globe like. So it is shown like this, spherical, right? So this is G actin, globular actin. This globular actin is mostly bound to ATP molecules, which are shown here with green dot. See the small dot that you see. These G actins, the ATP, it hydrolyzes and becomes ADP, and the G actins, they come together and form such kind of filament, okay, a fiber. Mostly it forms this double helical structure, just like DNA. You can see this one strand of actin, and then you have this another strand of actin. So they are double helical strands of globular actin. This actin, which now forms a fiber or a filament, is called F actin or filamentous actin. Clear? So this is how the actin is formed. Okay, it is made up of these actin, globular actin, okay, G actin, and G actin, they polymerize to form F actin. If you talk about the thickness, they are 6 to 8 nanometer in thickness. Okay, 6 to 8 nanometer. Where do they occur? So they are present below the plasma membrane. The red portion that you see here, okay, if this is a cell, then just below the plasma membrane, you can see there is a lot of red coloring. Okay, this is all because of microfilament. Microfilaments are also connected with spindle fibers, endoplasmic reticulum, chloroplast, and other cell organelles. Okay. Now, functions of these microfilaments. The first function is cytoplasmic streaming or cyclosis. Remember, we saw in that video how the chloroplast move around in a cell, okay, near the periphery. So this cyclosis, which is important for movement or even distribution of different molecules, is actually caused by activity of microfilaments. Okay, remember this. They help in stabilization of some membrane proteins. Just see this diagram. This is the membrane. Okay, this is your plasma membrane, and there are many membrane proteins. What you see here, the red colored filaments are actually made up of actin. Okay, remember we told it is present just below the membrane. So what is it doing just below the membrane? It is supporting some of these membrane proteins. Okay, you can see these membrane proteins are actually supported by actin. Microfilaments play an important part in change of cell form during development and differentiation. So while the cell is developing, while it is growing, this actin filaments or microfilaments play an important role during its differentiation and development. Okay, what kind of a cell it will make, the shape, the structures, all of it is decided by the actin. Okay, I'll give you a fun fact. Have you ever seen your thumbprints? Right, you have fingerprints. So these fingerprints, do you know how these fingerprints are actually made? They are nothing but they are arrangement of actin filaments. So from this, can you imagine how much they play a role in development and differentiation of cells? Actin filaments are the one which are giving you your thumbprints. Okay. Then microvilli. Remember the intestinal microvilli, right? So these microvilli are supported by the actin filaments. Okay, if it was only two membranes and there was nothing in between, these two membranes will collapse, right? So to avoid that, microfilaments or actin will be present in between the layers of this microvilli also. Fibroblasts are again cells you must have heard of in when you study animal tissues. Okay, can you remember areolar tissue? Areolar tissue having fibroblast. Fibroblast actually secrete different extracellular matrix, okay, like collagen, elastin, Fine. So fibroblasts are able to move due to plasma membrane undulation. Okay, undulation is wave-like movement. Have you seen a wave on a beach? So that movement is called undulation. Okay, so the plasma membrane undulation in fibroblast is happening because of microfilaments. Okay, so this means if you look at the fibroblast plasma membrane, it will look like a wave, okay, on the sea, and this is just because of microfilaments. Myofibrils, which are the contractile elements of muscle, have microfilaments. Remember this structure, okay, from your movement and locomotion chapter. You have this thick filament and then this thin filament. This thin filament is actually made up of actin. Okay, this green colored globular part you see is the actin. Okay, and on actin, the orange colored wire like thing you are seeing is tropomyosin, and this bead like structure is troponin. Fine, that is all secondary, but for us it is important to remember these actin molecules. Okay, this actin is very much important for contractile function of the muscles. Okay, then pigment granules, chloroplast, and other cell organelles are able to change their position inside the cytosol by means of microfilaments, right, which we described as cyclosis. So because of cyclosis, uh, the different materials are being able to diffuse properly, okay, evenly. Microfilaments help in formation and retraction of pseudopodia. Remember during where did we study pseudopodia? We studied at many places. We studied it during cell membrane. We also studied again when we were discussing lysosomes. So pseudopodia are these leg-like structures. Okay, these extension of the plasma membrane. So how will this plasma membrane, which is just a fluid small membrane, extend in this particular manner? It happens because inside it the actin, okay, so in the whole cell actin will be present in form of G actin, which is globular actin. Whenever there is requirement and you have to make a pseudopodia, this G actin will come together and start forming F actin, which you can see here, the strands. Okay, so wherever there will be increase in the length of F actin, you will see that the pseudopodia will move in that direction. Clear? So microfilaments help in formation and retraction of this pseudopodia. Whenever the function is done, then the F actin again starts disintegrating into G actin, and the pseudopodia it comes back. Clear? Microfilaments are responsible for changes in plasma membrane during endocytosis and exocytosis. Just as an example, you can see here there is a bacteria, and around it you are forming a pseudopodia to engulf it. Okay, this is phagocytosis again, but endocytosis and exocytosis are just the same. So in these two processes also, you can see these green colored fibers are actually actin fibers. Okay, so microfilaments are important in membrane phenomena like endocytosis and exocytosis. They are also important for cleavage furrow, which you see at the time of cytokinesis. Okay, in animal cell, there is a rubber band like structure, and when this rubber band it contracts, the two cell will divide. Okay, think of a balloon, and in the middle of the balloon you are tightening a rubber band. Okay, so as the rubber band will grow smaller and smaller, the cells will separate. Okay, so this is called the contractile ring, okay, or the cleavage furrow, and microfilaments are an important part of this process. Then you have cytoskeleton, the second type of filaments, which are called intermediate filaments. Intermediate filaments are 10 nanometer in size. Okay, so microfilaments are 6 to 8 nanometer. Then you have intermediate filaments, which are 10 nanometer. And then we will study microtubules, which are 13 nanometer in size, okay, in diameter. So you can see why it is called intermediate. It is between, its thickness is between the both of them. Okay, so these are solid filaments of about 10 nanometer. These are actually made of filaments, okay, made of fibers, not of globular proteins, but actual fibers, and the which are formed by variety of different proteins. They are of four types, okay. They can be keratin filaments. Keratin, you must have heard in hair, okay. Your hair are made up of keratin, your nails are made up of keratin, then the horns of, let us say, cows and buffaloes are made up of keratin, the claws of lion are made of keratin, then the horn of rhinoceros is also made up of keratin. So you can see where all this intermediate filament is very, very important, okay. Where we have studied in our in context to our chapter, we have studied it in desmosomes, okay. Remember the cell junctions. This is one type of cell junction, which is desmosome. So the desmosome is actually stabilized by these fibers, okay, and these fibers are nothing but they are intermediate filament of keratin. They are called tonofibrils, okay, in case of desmosomes. Then neurofilaments are intermediate filaments found in a neuron. Neuron is a nerve cell, okay. So here whatever the pink or purple colored filaments you are seeing, they are nothing but neurofilaments, and they give shape and structure, okay, support to the axons. Here this is the axon and the dendrites of a neuron. Same kind of intermediate filaments are seen in glial filaments, okay. So glial cells or astrocytes, which also you must have studied in this neuron chapter, okay, nervous system chapter. So astrocytes are supporting cells present inside the brain and spinal cord. So they also have intermediate filaments. Clear? If you see in a cell, where is this intermediate filament present? Then in this diagram, this is a fluorescent fluorescent light microscopy, okay, diagram, photograph, sorry. And in this photograph, all the green fibers you see are actually nothing else but these intermediate filaments. So here you can see it will be providing support to the nucleus as well, because it is thick around the nucleus and then it is thick around the cell membrane, okay, and it is present throughout the cell. In this diagram also you can see that it is present near the desmosomes, okay, which we just discussed. Desmosomes are the cellular junction, okay. So between the two cells, wherever there will be desmosomes, you will find such tufts of tonofibrils or keratin, okay. Now what do they look like? So here the monomer looks like a fiber, okay, it itself looks like a fiber. Remember, in case of microfilaments or actin, the monomer was actually globular. In this case, the monomer itself is like a filament. Then two monomers, they come together to form a dimer. Two dimers come together to form tetramers. And such 8 sheets of tetramers, okay, you can count 1, 2, 3, 4, 5, 6, 7 and 8. Such 8 sheets of tetramers, they come together and form such kind of a carpet like structure. And when you roll the carpet, you form this rope like filament bundle, okay. And this bundle is very, very strong. It is super coiled, it is very strong, and so it provides much support to the cell. Clear? Can you understand? Just like a carpet, these tetramers are arranged, and when you roll the carpet, you get such kind of a filament bundle. They're absent in unicellular eukaryotes and evolved only in multicellular eukaryotes, okay. So if you talk about unicellular eukaryotes, they will not have intermediate filaments. They are present only in multicellular eukaryotes, because remember the reason, they are important for connections, okay, the junctions between the two cells. In unicellular organism there is only single cell, so where will it form a connection? So there is no need for intermediate filament. While in multicellular organism you have multiple cells, and so these multiple cells should be connected, and they are connected by desmosomes, and these desmosomes are supported by intermediate filaments. The filaments are cross-linked with one another and also with other cellular structures. Functions of intermediate filament. Um, they form in nuclear matrix. It forms the it is mainly formed of intermediate filaments. Okay, here then it provides support to all biomembranes. Okay, not only the cell membrane, it provides support to the nuclear membrane. It provides support to other membranes as well. Okay, including plasma membrane and nuclear membrane. A lattice of Desmin filament provides support to contractile units of sarcomere. Okay, when we will discuss sarcomeres, you will notice there is a filament, another filament called Desmin. This Desmin is actually made up of intermediate filaments. Okay, it is a type of intermediate filament and it supports the sarcomere. Okay, desmosomes are supported by tonofibrils. Then intermediate filaments maintain integrity of epithelial tissue. Okay, your epithelium, its shape, size, and the integrity. Okay, its original shape will be maintained because of intermediate filaments. Keratin is deposited in the skin cells and this provides protection against abrasion. Abrasion is basically friction. Okay, so on your skin cells, in your hair, you have a lot of keratin, and this keratin is actually present to protect you, to protect your cells. Okay, so the uppermost layer of your skin is dead cells, and these dead cells have a lot of keratin. Clear? Now we talk about the last part of cytoskeleton, which are microtubules. Okay, they are unbranched. So there is no branching. They are hollow. They are tube-like. Okay, they are straw-like. So hollow tubular protein filaments. They develop on specific nucleating regions. Matlab, they cannot be seen anywhere and everywhere. There should be some point where they start to aggregate. Okay, and that point is called nucleating region. Clear? They develop on such specific nucleating region and can undergo quick growth and dissolution. To understand this, let us just first understand the a parts of a microtubule. Okay, it is made up of two types of same protein. Okay? So the protein is called tubulin. Write it down. The protein is called tubulin. Okay. Now this tubulin protein has two varieties. One is called alpha tubulin, which is shown here in purple color, and the other one is beta tubulin, which is shown in golden color. Okay, so you have this alpha and beta tubulin. Sorry. So this alpha and beta tubulin form a heterodimer. Hetero means different, di means two, and mer means two different monomers have come together. Okay, and they are forming this structure. So these are the tubulin heterodimers. In this, the beta portion, okay, the beta tubulin has the site where ATP can bind. Okay, remember the G actin, it had the site where ATP can bind. Same way, here the beta tubulin is a place where ATP can bind. Now what happens is when that ATP, it gets hydrolyzed, it forms ADP and phosphate, and its site is free for attachment. In such case, these heterodimers, they come together to form this one string. Okay, one necklace like structure. This is called protofilament. Okay, so heterodimers, they come together to form protofilament. Wherever there will be beta tubulin. Okay, so here the beta tubulin is seen on the right side. That is called positive end, meaning you can add more heterodimers on this side. And wherever there is alpha tubulin present, that is called the negative end, because there is no ATP binding site or binding site of any type. No heterodimers can be added on this side. Okay, it can only be added on the positive side. Clear? So whenever it has to grow, if let us say the microtubule has to grow, then it can only grow on the positive side. Clear? So this is protofilament. Many such protofilament. How many? 13. Okay, 13 protofilaments come together and they form this circular straw-like structure. Okay, which looks like this. Count these numbers, you will find 13. Okay, so 13 protofilaments, they come together to form this hollow tube-like structure, and this hollow tube-like structure is now called microtubule. Clear this much? Its diameter is 25 nanometer. If you talk about the diameter, it is 25 nanometer. It is formed of 13 helically arranged strands of protofilaments. These are the protofilaments, okay, made up of alpha and beta tubulin protein. Now there is a dye called colchicine. This colchicine, it prevents spindle formation. It will not allow this positive end to grow. Now because these microtubules are important, okay, they are present as spindle fibers during cell division. So if colchicine is added, the spindle fibers will not form. If the spindle fibers do not form, the chromosomes will not separate. Okay, so colchicine is a dye which is used to halt, or which is used to stop cell division, and then you can easily look at the cells under a microscope and study them. Okay, you can fix the cells in their position of cell division if you use colchicine, because it inhibits this microtubule extension. Clear? Right, so here is polymerization. On the positive end, you will have more and more heterodimers which can come and attach. Okay, so this will increase the length of this microtubule, and when this microtubule is no longer required, what will happen is it will start disintegrating. Okay, it will start falling apart. The heterodimers will start coming out. Okay, they will start separating into these smaller monomeric units. Okay, so when it is having GTP, that GTP wala heterodimer can polymerize, but when it has GDP, okay, so this GDP wala unit cannot polymerize, it will separate. Okay, so this is what happens. Catastrophe it is called, but that is not important for you. Anyway, so this was just a diagram I put to make you understand what is polymerization, how it is happening, and how it can even be broken down. Okay, this is how a microtubule can be broken down from a place where it is no longer needed. Now, functions of microtubules. They are structural components of spindle fibers. Okay, so spindle fibers, I told you, they are helpful in separating the chromosomes during mitosis or meiosis. Then you have chromosome fibers. Remember the kinetochore? If not, we will be starting with that chapter. So we will study. Okay, so these chromosome fibers are also made up of microtubules. Centrioles are made up of microtubules. Basal body of flagella and cilia, as well as the flagella and cilia, are all made up of microtubules. Okay, so here what you see, this is a normal nerve cell. This is a dendrite and this is an axon. So even all of these portions will have microtubules. Then this is during cell division. You can see these are the centrioles, and from the centrioles the orange colored fibers are nothing but microtubules or spindle fibers, okay, which are made up of microtubules. Then in ciliated epithelial cells, where there is presence of cilia, you also have at the base, okay, you have at the base microtubules, okay. And for example, in this RBC also, red blood cell, you have microtubules which are present in the periphery. Okay, they provide rigidity and shape to some parts of the cell. They control the shape of wall-less cells and nucleus. Okay, so the cells of animals, okay, which do not have a cell wall, their shape is maintained by microtubules. They take part in intracellular transport. Okay, inside the cell, whatever transport is happening can be made possible because of microtubules. Remember, I had told you that microtubules are like highways, and on these highways there is movement of vesicles. Okay, we will see that in the next or coming slide. Okay, in plant cells, the microtubule control the orientation of cellulose microfibrils. Okay, remember the cell wall. So in that cell wall you had cellulose microfibrils. So these cellulose microfibrils are maintained by microtubules. They help in movement of nuclei during cell division. So this is what is meant by intracellular transport. This is your microtubule. It will act like a highway. On this, remember that guy who had two shoes and he was moving around with the vesicle, right? So same thing, this guy will use this um, microtubule like a highway, and it will transport the vesicle wherever it is required. If there is need for exocytosis, this vesicle will be led towards the plasma membrane. If this vesicle is from endoplasmic reticulum, and if it should go to the Golgi body, then this microtubule and the motor protein, okay, this is called a motor protein. So this guy on the motor protein will help uh, reach this vesicle to the Golgi body. Okay, now let us see the video. Okay, this is the bottom part is your microtubule, and as the ATP get hydrolyzed, the protein moves ahead, and attached to this orange colored protein is your vesicle. Okay, the big thing you see at the back is your vesicle, and this is actually how the vesicles are transported inside the cell. They take part in separation of chromosomes during anaphase. Then the place of cell plate. Now the cell, because of the cell wall, the plant cell cannot have a cleavage furrow. So how will it decide where the two cells should divide? So there is presence of these microtubules in between as phragmoplast, and wherever this phragmoplast will be present, the first what you can say middle lamella will form there. Okay, if you can understand this, well and good. If not, do not worry, we will be discussing this when we cover the next chapter, which is cell division. Okay, cell cycle, cell division. So basically this cell plate which divides the two plant cells is decided where the cell plate will form. That is decided, sorry, by microtubule. Then distribution of microtubule determines the cell polarity, and microtubules help in movement as part of flagella or cilia. For example, this here is a unicellular organism, protist, okay, which has a lot of cilia, as you can see. When you take a cross section, this is that one cilia. When you take cross section of that one cilia, you will find such kind of a structure. This structure has these rings, okay, and these rings are nothing but they are microtubules. Okay, so microtubules are helping in movement of such flagella as well as cilia. Flagella will also have the same arrangement of microtubules. Okay, so we will discuss flagella and cilia from the next class. Till then, I hope you had fun and you are reading, solving questions. Okay, see you next time.